Hello, and welcome to this wonderful joint seminar between Mission Bio and BioLegend, in which we'll showcase the new product launch to interrogate DNA plus protein and unravel the complexity of cancer with single-cell multi-omics. With us today from Mission Bio, we have Senior Product Manager Jim Flynn, and from BioLegend, we have Product Manager Adnan Chowdhury. So with that, I'll hand it over.
Thanks, Kelly, for introducing us. Very excited to get going today. I see we have participants joining us. We're going to talk a little bit today about multi-omics and how that could be utilized in unraveling the complexity of cancer. So with that, I'll just start with a brief introduction to the Tapestry platform. We'll then move into talking about some of the new products and the new workflows that we have available in partnership with BioLegend. And then finally, we'll have Adnan talk a little bit about some of the product from BioLegend and how that's going to be utilized on the Tapestry platform. So just starting off, giving a little bit of background about what Mission Bio does and who we are. We are very interested in moving precision medicine forward. And what that means is that we are very focused on helping researchers and clinicians understand what's going on a single cell level when a patient has cancer. And primarily we've been focused on the heme-oncology, leukemias and lymphomas types of cancer, but we're rapidly moving into other cancer types that are solid tumor based. We've started with the single cell DNA solutions. Now we're moving into other analytes that are protein and things like that. So we'll be talking about that today. So just a little bit of history about Mission Bio. We are a startup company that's been focused in this for several years now. We've sequenced over 10 million cancer genomes at the single cell level. We do about 34 custom panels. It's probably a much higher number. We're doing about 50 to 60 per year. We have 360 users on our Tapestry portal, and we're in about 1/5 of all cancer centers have adopted the platform, and that continues to increase over time. So that's kind of a little bit of background about what Mission Bio does. Why do you need to know about information of the genome of a cancer cell at the single cell level? And we can show you why this is in this schematic here. You can see that we have a cell population of normal cells. As you age, you continue to acquire additional mutations. And unfortunately, you may hit upon a mutation, which is an oncogene or a gene which drives a particular type of cancer. And then once you have that, that cell begins to divide in an uncontrolled manner. And then as that happens, those cells begin to acquire additional mutations on top of that. And so the cancer evolves, and it's in an environment where it's under selective pressure. So maybe you treat the cancer with a particular drug that may kill off one of those clones of cancer, but then other types or other clones which have different sets of mutations may evolve and may begin to be the dominant clone that happens. So it's very much an evolutionary process. So you want to understand at the single cell level which one of those cells is present and what are the mutational profiles within those sets of cells. And the only way to do that is at the single cell level. If you mashed all of these cells in the schematic up, you'd get some VAF or some varying allele frequency, but it doesn't really tell you which sets of cells and which have which mutations. The other piece of DNA sequencing is that you want to resolve that clonality. You want to understand what the clonal substructure is of each one of those. You also want to understand the lineage or how that cancer is evolving over time. And so you can begin to put together these kind of family trees or lineages of the cell populations. In addition to that, you want to see over time with different treatments, what's happening to those tumor cell populations. So you want under a particular treatment or selective pressure, which are the clones that return or are more persistent in that patient. And that will help you understand what is the right treatment going forward for a patient. And then finally, you want to understand which sets of mutations which occur together are most deleterious to a patient, and that will also help guide the treatment that the patient gets. This is a slide that's just highlighting what I just mentioned, which is the difference between bulk sequencing and single-cell DNA analysis. If you do bulk sequencing, if you have this image of the test tube, you can see that there's cells that have different mutational profiles. And this is just highlighted as the red dot and the blue dot, which represent two different mutations. And as you can see, some cells have just one mutation or the other mutation, and some sets of cells have both sets of mutations. If you do bulk sequencing, you're really just assaying what are the sets of DNA in that cell. You're not looking at it a cell by cell basis. So you'd get some answer that tells you, well, half of the cells carry mutation A, half of the cells carry mutation, or sorry, 40% of the cells carry mutation A, 40% carry mutation B. But what it doesn't really tell you, and what I'm showing here on the bottom, is that actually 20% of the cells have just one mutation, 20% have the other, and 20% have both of those mutations. And having the presence of both of those mutations in a subset of those cells may be a very different or more aggressive form of cancer than if you had just mutation A or mutation B. So understanding that on a cell-by-cell basis really gives you a deeper level of understanding that you would not get from bulk sequencing. This is just another way of looking at it, is if you have one sample, you then get millions of reads from that set of genomes, and it will give you a profile on the right, which says, oh, okay, I have an IDH1 mutation and an IDH2 mutation. But what we're showing on the bottom was with Tapestry is you do that by a cell-by-cell basis across thousands of cells. You still get millions of reads, but at least we've barcoded each one of those reads, and we can map that back to an individual cell. And then we can bioinformatically then dissect that, which reads came from which cell. And now on the right, we're showing you that you can actually see, I do get IDH1 mutation, IDH2 mutations, but now I understand which sets of cells have those mutations, but then I can also observe additional mutations which would be missed by bulk sequencing because we're doing it on a cell-by-cell basis. And so you get a much more sensitive or deeper understanding of the mutational profiles from that patient across thousands of cells. Just showing now what we offer. We are a complete end-to-end solution. So we offer the assay. So we have all the reagents that are contained in our core reagent kits. And I'll talk a little bit more about what we've updated with DNA and now protein being added as an additional analyte. We also offer the Tapestry instrument, which is a microfluidic device. It houses a microfluidic cartridge that you put all the reagents and their samples into, which then barcodes and isolates each individual cell. And then we also have all the bioinformatics and analytics in our Tapestry pipeline and our software. We have desktop software. We also have Python code-based packages for more advanced users who want to create additional visualizations or dig a little bit deeper into the data. So we have all of the pieces to complete this workflow offered by Mission Bio. And now with our partnership in BioLegend, we have antibody tagged, oligo tagged antibodies, which you can now add into the workflow as well. And it gives you a protein as well as this DNA information. So just now highlighting what makes Mission Bio different from other microfluidic platforms is that we have a unique two-step workflow. And so if you start on the left of the workflow, the first phase is we run it through the microfluidic devices. You input your cells, and that goes into an oil emulsion droplet. And in that first droplet, we also introduce a proprietary cocktail of proteases. And then when you have that cell now encapsulated with the protease, we can lyse the cell in a very controlled fashion within that droplet. And that allows us to access the genomic DNA by stripping all the protein off of that. And that frees up the genomic DNA for a highly multiplexed PCR reaction. So in the second step on the right side, you can see we then have this now droplet goes back into the microfluidics and goes into a second droplet, which introduces all the PCR reagents and the barcoding beads. And that's a critical step is that now we've accessed the genomic DNA. We can now introduce our barcoded bead and reagents, and then we can amplify up specific regions of the genome. And then, so this is a very targeted assay, and it allows you to ask just the regions of interest to a particular type of cancer. What are the mutational profiles within those regions? So we can do anywhere from 20 amplicons up to about 1,000 amplicons to assay across the genome in a targeted fashion. And then what results is you get an Illumina-ready sequencing library, and then it's a standard Illumina sequencing. And then, of course, on the other end, we have all the bioinformatics to deconvolute the barcodes and then read out what the mutational profiles are of each cell. This is showing you how you would utilize that information. So we have single cell heterogeneity of MRD, which is minimal residual disease. So it's showing you here, these are called fish plots, and it's really a representation of the patient's history over time. So you see at diagnosis, we have in red, it shows you what is the dominant clone that's present. Is it normal cells? Is it a cancerous cell? What are the mutational profiles within that? Are there different clones present? And then you can see at diagnosis, you have one profile. You will put that patient on some sort of treatment. They will go into remission, and you can see now on the left side that you get mostly wild type or normal cells because the patient's now eradicated most of the cancer. But then you can see in a monitoring phase, when patient's in remission, you can see we do detect, oh, there's 6 cells, and the patient has, in those 6 cells, this mutational profile. We can see they have a quadruple mutant clone present, shown. And then you see that that's the clone that actually comes back when the patient relapses. And so at relapse, you also want to understand what is the right treatment for that patient. So in the middle, we're showing here at diagnosis, they had a dominant clone shown in red. We do detect some other minor clones present. And then you put the patient on treatment, they're mostly wild type cells, but then you do detect there's a few cancerous cells, and they do have a different mutational profile than when the patient started. So at that point, the clinician may say, oh, I want to change the treatment that this patient goes on to. And then finally, on the right side, we're showing you that you may have cancer, but you also may have a background of what are called chip clones, or cells that have potential to become cancer present in the patient, but that's not the entire story of what's happening in that patient. So by doing single cell sequencing, you get a much deeper profile of what's going on in a patient, whether they have normal cells, precancerous cells, or they have actual full-on cancerous clones going on in that patient. So it gives you a very deeper profile than you get from bulk sequencing. How is this useful? Well, now we've had a number of publications out there. This is actually some schematics from that, showing that patients that have a similar degree of clonal richness at diagnosis, but a lower clonal diversity, may have a longer relapse-free survival. So understanding how complex the cancer cells are, you can imagine if you have one type of clone, that may be manageable to treat that clone. But if you have a diversity of different types of cancer mutations going on, it's much harder to pick one treatment that may work across all of those clones. And what our customers have actually shown is that the higher diversity has a much worse outcome potential of being disease-free for a longer period of time. So if you understand that at the point of diagnosis, you can set expectations much better than if you had a single clone, let's say. So this is quite powerful for people that are running clinical trials to understand which therapeutic strategies are going to be best and which will be most effective at each given time point in the patient's treatment. I did mention we have a growing number of publications. So we've done this across now AML. We have a number of publications out there. We've had people using this in clinical trials as a kind of secondary piece of information to understand what's happening with the patient. So if the patient goes on therapy and they develop resistance, they've been using the Tapestry platform to understand why that resistance is happening by delving deep into their mutational profiles. They've also expanded into additional indication, whether that be multiple myeloma, ALL, as well as others. And customers have actually been branching out into a number of additional applications, such as virology or cell and gene therapy, but it's not the focus of this talk, but we do have additional applications which people are using the platform for. I also just want to highlight that we've had some partners such as LabCorp who have adopted the platform, and they're using this in early phase trials to evaluate resistance mechanisms. And they're finding that the sensitivity of the Tapestry platform is quite useful in understanding which patients to put in clinical trials, which not, who has resistance profiles, which explain why a drug does and does not work for a particular patient. So I just described a bunch of things that are going on with just looking at the DNA only of a patient. And what we really want to talk about today is that we really want to expand into multi-omics, which is not just a single analyte, whether that be DNA for particular SNPs or SNVs that are present in the patient, or copy number changes that happen in their genome. It's really, to truly understand cancer, you need to integrate multiple pieces of information. And so in this cartoon, I'm just showing you that if you can take multiple threads of information and integrate those signatures, that gives you a much better profile or more effective biomarkers to select the right therapeutics. And if you do this at the bulk level, you kind of collapse all of that information and it'll give you some profile, but it doesn't tell you what's happening on the individual cell by cell basis. And as we've shown with DNA, the profile can be quite complex just looking at one analyte. So you want to do it at a single cell level, understand on a cell by cell basis what's happening and across multiple analytes. And that gives you a much better information of linking the genotype of a particular cell and then what results as the phenotype from that same cell. And so that will uncover unique disease signatures for personalized therapeutics. So this is, again, kind of a different way of looking at that, is that you may have precious clinical samples from a patient, and you may want to have multiple pieces of information. So you might do NGS to get at the DNA mutations. You might do flow cytometry to understand what are the cell surface markers, or what's the different sets of cells that are there based on the protein expression. You may want to understand copy number changes either on the single cell level or by array. But then you have to begin to stitch all those data sets together. They're completely different platforms. There's no integrated solution that will allow you to say on a cell by cell basis, here's the cell surface markers, here's the mutational profile of that cell, and then do that across thousands of cells. So what we're showing is that you have to, if you do it separately, you have to make assumptions when you have to assume that this cell, I think this cell has this set of surface markers because it kind of matches this set of cells which have this mutational profile. But what we're highlighting on the right is that you can take that same clinical sample, you can run it through the Tapestry platform with our multi-omics workflow, and then you have an integrated data solution that will analyze all that information and integrate all of that information of the mutational profile of the cell, plus the cell surface markers, plus copy number changes that may be happening in particular chromosome arms or genes. And you can integrate all information with a desktop software that will do this for you. So now this is what we are talking about today, is what we're launching is a complete workflow for single cell multi-omics solutions. And so we have now, with our partnership at BioLegend, is that we can offer the cell surface protein antibodies with oligonucleotide tags. We have the instrumentation and we have the software all available in a single workflow to analyze cancer samples. So these are some specifications around it. We have our platform, and it allows you to do targeted single cell DNA and protein profiling. Both we have standard catalog panels that will do this for certain disease indications, but also we have the ability to customize any panel or sets of antibodies to assay the cell surface proteins. And so we can do this across hundreds up to 1,000 different genomic loci. We can do chromosome and gene level copy number change detection using our algorithms. We can do this, and typically, depending on the sample quality, it's going to be from anywhere from 4,000 up to 10,000 cells per sample per run. It's very high sensitivity, down to 0.1% of the cell population. You can observe a mutation, and as well as phenotype that with the cell surface antibodies. As I mentioned, we can customize and we have different options to do that. We have software that you can go and sign in to our website right now, and you can design your own panel. Or we have a team of bioinformaticians that will build a panel for you if you have very specific requirements. And all of this workflow has been optimized to work on the typical cancer cell samples, which are PBMCs or bone marrow derived cells. We also can work with nuclei, absent the protein, or cell lines. So all of those are available. This is just highlighting our kind of product portfolio and how that works for Multi-Omics. You can see in the purple box here, Mission Bio offers our DNA and protein core kits. So it offers the reagents to do the cell staining, as well as to do the highly multiplexed DNA portion of the workflow that will amplify up the specific genomic regions of interest. And all of that, when you purchase the reagents, comes with the cloud computing and access and analysis. So we have a cloud pipeline that you can upload your Illumina sequencing libraries to, and that will deconvolute the barcode sequences and then essentially sort the reads based on a cell-by-cell barcode. To those core reagent kits and our cloud computing, we then add in our catalog DNA oligo pools or custom DNA oligo pools. So you have the option to buy catalog or customize if you so desire to do for each particular sets of cancer mutations. And now with our partnership with BioLegend, what Anand will talk about is we have the TotalSeq D Heme Oncology panel, which is a 45-plex pre-titrated, pre-optimized set of antibodies with the most common heme oncology cell surface markers. So it allows you to phenotype each cell based on these 45 markers. BioLegend is also offering custom antibody conjugation. So if you want to add to that panel of 45 or just develop a completely unique set of antibody conjugates, that's available from our partnership at BioLegend. And then on the right, I'm showing you we have the complete software and bioinformatics solution, which is our Tapestry pipeline, which is basically a cloud-based pipeline you can upload your library to, and it will deconvolute the data. Once you get that, you will get a data file back, which can go right into our Insights desktop software, which is very easy to use, allows you to explore the data. That is freely available. You can download it and play with some data sets from our website if you want to do so. Also freely available is our Mosaic package, which is a Python code package, which will be available on GitHub for anybody that wants to try out or play with some data sets. Those are available to download. Now I just want to highlight the power of Multi-Omics. So this is showing you what you can do with the platform, essentially. So this is just looking at a mixture of 4 cell lines. So it's a sample experiment just to highlight what you can do. So these cell lines tend to be kind of homogeneous and it's the same across them. So if we're looking in A here, what we're showing is this 4 cell lines which were mixed together roughly equal proportions. Is if you look at the mutational profile across a couple of sites, you can see that in the blue cell line, I can clearly see some mutations that differentiate them from the red and the purple. And then the green cell line, it can also differentiate based on genotype, but I can't genotype the red and the purple data set. They look identical from their mutational profile perspective. And so if I look now in B in the bottom and I look at under SNVs and CNVs, I can't really distinguish those 2 cell populations. I can clearly distinguish the blue from the green based on mutational profile. Similarly with CNVs, I can't really distinguish super cleanly the blue from the purple cell population just based on that CNV information. And then from a protein perspective, I'm showing you in A, is that I can sort of distinguish populations, but not in a very clean fashion. I can't cleanly dissect apart those different cell populations. And if you recall back to the slide I just showed you previously, that's how people would basically do that if they were not doing a multi-omics workflow. You'd take different sets of information, you'd run different assays, you'd begin to sort of dissect out different cell populations. But then when you go to integrate that information, you have to make some assumptions about, does this cell really fit in with this population? You can't do that on a cell-by-cell basis. But with the Tapestry platform, you get all of this information from the same cell, from the same sample. And so what I can do now, I'm showing you on bottom of the three right population of the t-SNE plots, is that if I combine CNV and protein, I can begin to start separating those populations out. Similarly, if I had just the SNP information or SNV information plus protein, I can begin to dissect those populations. And then on the far right, it's showing you if I combine all of those pieces of information on a cell-by-cell basis, I can very cleanly dissect apart what's happening in this population of cells. And I can clearly distinguish all 4 cell populations from each other. And I just want to highlight, this was just done with a very small panel of 127 amplicons using just a very small subset of antibodies. So just 6 antibodies and looking at 127 amplicons across 20 genes, I can cleanly dissect all of those cell populations from each other. And so that's really the power of multi-omics and how it can be utilized to dissect apart cancer cells. I just want to highlight a couple updates to our product offering. This is now what we offer for software. So I mentioned you can create your own custom panel. You can go to our website and create an account and sign into Tapestry Designer and begin to construct a panel. It's very easy to use software. And that will design specific amplicons to target regions of interest, either across the whole gene, or if you have particular SNVs you want to target or copy number changes, you can build a panel in our software. It's very easy to do. It's free for anybody. As we mentioned, when you buy the reagents, you gain access to our Tapestry pipeline. So this is a new update for us. The Tapestry pipeline now will support multi-omics workflows. So if you have your sequencing library for the protein information and for the DNA, it will combine all of that information and deconvolute the cell barcodes. And then finally, to create all the visualizations, or many of the visualizations I showed you, we have a desktop software that does that. And again, it's free to download and you can play with data sets. And that will help you understand the single cell data. This is just showing you our Tapestry Designer. You can go in there, you can explore our catalog panels and understand exactly where the variants are targeted and visualize that. You can also download all the design files and the data sheets that are available there, as well as build custom panels. And we do offer that for human and for mouse. So if you have a mouse model system, it's supported in there if you would like to build panels. We also have some publications showing both human and mouse projects that have been useful to use our design software. On the right, it's just showing you our design software is optimized based on wet lab experimentation, and it continues to improve over time, but it's just showing you we have a very high degree of uniformity and amplicon performance in our software. And uniformity is important because you don't want all the reads going to one amplicon. You want a very even amplification. So we have highly optimized chemistry to do that. And we get very good uniformity, which translates to saving you sequencing dollars on the other end. Our new Tapestry pipeline is our variant calling software. This new pipeline is now optimized for multi-omics, and it has a bunch of metrics that will come out now when you run our pipeline. So after you sequence and you run on a pipeline, it tells you exactly how well your experiment worked, as well as the quality of the cells which are called. That's all available. And again, included when you purchase the reagents, you gain access to that pipeline. And then finally, I want to highlight a major update to our software. So previously, our software called Insights only did DNA visualizations. This now fully supports multi-omics data for both DNA mutations as well as protein. And again, you can go and you can download this and try it out, and it will have access to multi-omic data sets, so you can begin to see what you can do with the software. And if you want even more visualizations or ways to analyze the data, you can gain access to Tapestry Mosaic, which is available on GitHub, which is a Python code-based package. This is just some examples of that Insight software. Again, you can take data sets, you can create t-SNE plots or UMaps. You can then color in different pieces of information. If you want to know just one particular marker, where is that marker, that protein marker expressed in which cell populations? With just a few clicks, you can begin to create all of those visualizations very easily. And on the right, we're just showing you now a heat map across cell populations. And you can see we can very cleanly dissect out, based on their cell surface profile, different subsets of cells, was naive T cells, memory cells, NK cells, B cells. So you can do that across different cell populations, as well as understanding if there's cancer cells present. So this is normal PBMCs I'm showing you in the heat map, but if you had a cancer cell population, what does that cancer cell population look like from a cell surface phenotype, as well as understanding on the right, you can see the kind of blue-gray bar, the mutational profiles within each of those subsets. We've been working with a number of our key opinion leaders and our kind of labs we work very closely with as collaborators. I just want to highlight what I've done using this DNA plus protein workflow. There's been 2 recent Nature publications which come out. The first one being Ross Levine. We work quite well with their lab, and they were interested in understanding what happens as the cancer evolves from a precancerous state, or you may have a clone that has acquired some mutations, but it's not really evolved into full acute myeloid leukemia, and how does that evolution process happens. So they used our multi-omic approach, and they understood all the genotypic changes that were happening, but then they also wanted to understand the cell surface changes that are happening in those cells. And that's very important because from a cell surface perspective, many of the drugs which are given to patients to treat them will target a particular cell surface marker. So it's very important that they understand what changes as the cancer cell evolves. So you can see here, they can identify different clones, which are kind of shown in this, this reddish beige plot. You see different clones are present in these patients, and they acquire these heterozygous or homozygous mutations. In particular, they had a number of KRAS and NRAS mutations which became present. And you can see that in those patients, the wild type patients had a particular cell surface profile. But if you acquire these RAS mutations, you get an increase in CD11B expression. So the cells do start to look quite different from a phenotypic perspective. And so they just need to understand that profile. We also, one of our collaborators at MD Anderson also utilized this workflow, and they were looking at AML surface phenotypes, but also genotypically information. And so this paper from Koichi Takahashi's lab, they wanted to understand how that cancer branches or evolves over time. So you may start with one clone of cancer, and then it starts to acquire additional mutations, and that may become the new dominant clone or subclone. And then so we can see in the figure I'm showing on the right, you see we have this blue clone which acquired a single TP53 mutation. That cancer further evolved to have a second clone which now had a double mutation. And if you look in the plot in the bottom, showing these kind of bubbles, you can see the first 2 rows, they have roughly similar cell surface marker expression, which they're showing there. And then you see this small red clone, which then became a triple mutant clone that had 2 TP53 mutations and then acquired a KRAS mutation. The cell surface profile or the phenotype of that cell, very different than the 2 previous. So you can imagine if I was targeting a particular drug to a cell surface marker, and then all of a sudden you get a 3rd mutation, and then that profile looks completely different, then it gives me a much better understanding if I'm targeting a particular marker, how effective it will be for the most aggressive clones within that cancer. So I just covered kind of the use case for that. I'm happy to answer any questions at the end of the talk, but I want to turn it over to our partners at BioLegend and have them talk a little bit about all the work that they've done to develop some new products that work on the Tapestry platform. So with that, I'll turn it over to Adnan.
Thank you, Jim. I'd like to thank Mission Bio for inviting me to this webinar and for everyone for attending. Next slide, please. I'm super excited to announce or talk about our partnership with BioLegend and Mission Bio. This partnership was actually formed to bring pre-titrated cocktails to add protein expression to single-cell data. As Jim has established, Mission Bio is a leader in providing genotyping information at single-cell resolution through their Tapestry platform, which can then identify copy number variation and single nucleotide variation together using a targeted amplicon approach for genotyping. And he's just mentioned that they have a great robust solution for identifying or integrating DNA and protein analysis together. And what BioLegend brings to this partnership is that we're a leader in single cell based analysis. We actually have the largest portfolio of single cell antibodies used for single cell studies. And we're actually the only company right now offering a pre-titrated lyophilized cocktail. And so what this partnership has led to, next slide please, is our Total-Seq D Human Heme Oncology Cocktail version 1.0. Now, what's great about this cocktail is that it has 45 antibodies. If there's one thing I want you to take from my portion of the talk, is that you have 45 antibodies able to phenotype and subdivide and identify subpopulations of cells. On the right, I'd just like to bring your attention to these are just an example of UMAPs with various major markers used from this panel. As you can see, we have like CD3, CD4, CD14, etc. And in the lowest right of the panel, the cell type, we can identify these cell types by T, B, NK, and monocytes. And this can be further divided into other cell populations and subpopulations. The great thing about this cocktail, it comes pre-titrated and lyophilized. The pre-titration eliminates the need to mix your own antibodies. Mixing 45 antibodies can be quite a chore, and it'll eliminate batch effects. And the titrations also eliminates the need or resources to titrate your own antibodies. These are ready to go once they're reconstituted. This is actually formulated for applying to 1 million cells, and they come in a pack of 8 reactions, so you can do 8 samples with this cocktail. The part number is listed here, and you can order them on biolegend.com. And so with this cocktail in our partnership, it actually integrates seamlessly into the Tapestry workflow. If you next slide, please. And so all you need to do is stain your cells with the Total-Seq D upstream on the very left, and then it just goes right into the Tapestry workflow. And so you encapsulate your cells on Tapestry, and then you release the DNA and protein from within the droplets. Then these droplets are then barcoded in the next panel. And then finally, the targets are amplified and you get your library prep and you can do sequencing and analysis. Next slide, please. So this is just an example of what you would get when you purchase the product. This is a very simple cocktail to use. On the bottom, you can see there's a reconstitution volume of 60 microliters. You'll then just use 50 microliters of that with your 1 million cells blocked in 50 microliters of stain, leading to a total staining volume of 100 microliters. Next slide, please. On the other side, it just has detailed instructions on how to actually reconstitute and apply the cocktail to your cell population. And at the very end, it'll direct you to the Mission Bio website to further take your sample from after they've been stained with Total-Seq D into the Tapestry workflow. Next slide, please. This is actually a list of all the markers found in the cocktail. It actually has 45 antibodies, 42 of which are markers. And then at the bottom here, we have 3 isotype controls. This list can be found on our website, which will be given at the end of the talk. Next slide, please. Yeah. One thing I wanted to highlight is that these cocktails come pre-titrated. And so these pre-titrated cocktails give you optimal performance. And what I mean by optimal performance is not only that it's mixed very consistently, but what I want to bring your attention to is the right. Here we have resting PBMCs, and they're stained with a panel that has not been optimized or untitrated yet. And I want to bring your attention to the middle of that panel, CD54. In CD54, you can see we can identify the population that is positive for this, but it actually is consuming 8.5% of the reads. And then afterwards, on the right side of this panel, you can see that now with titration, we've actually reduced that percentage of reads by about 10-fold. And so this enables you to get the same result of identifying a positive population, but eliminates the need of excess sequencing that's completely unnecessary. Next slide, please. And so finally, just as I mentioned, I would like to mention that the 45-plex panel, if you don't have your favorite marker in there, if you want to build your own panel, we do have custom solutions available, and these can be requested from Mission Bio or from us at BioLegend. You can just use the contact information below. And if you would like further information, here's a link for the TotalSeq-D website at BioLegend. And again, this link will be provided in the chat. Next slide, please. And thank you very much. I'll let you guys take it away.
Yeah, thank you for attending. We're happy to answer any questions that you guys may have. You can either, I believe, ask in the chat. I don't know if people can ask live.
Yes, so thank you very much, Jim and Adnan, for a wonderful talk on the new DNA plus protein product. And we definitely have a few questions from the audience, so we'd love to get your expert opinion. Although this is a softball, so I'll ask it first. Do you have a tapestry installed in Germany? Do you have customer support in Europe? And are there sales reps in Europe?
Great question. Yes, we do have sales in Europe. I believe we do also have one in Germany. So please do reach out to us. Either my email is Flynn, F-L-Y-N-N @Mission Bio, and I'm happy to put you in contact with the right people. Otherwise, you can use this email address shown on the bottom here, info@Mission Bio.
Great. And then about BioLegend, are you in countries around the world?
Yeah. We're available in Europe as well as Asia. Great.
So how do you barcode individual cells? Can you explain a bit more about how that process works and how to eliminate doublets?
That's a great question. Yes, if I showed you that microfluidic kind of workflow. So in the first phase of the workflow, you encapsulate a single cell into that droplet. And then in the second phase, we introduce a barcoded bead. So each bead has a unique barcode on it. That's introduced with the reagents and now the lysed cell. And so when you have that barcoded bead, it has oligos attached to it. Through a triple primer PCR, we introduce a bridge sequence, and that attaches to the specific amplicon. And so when you PCR all of that up together, it introduces this cell-specific barcode, plus using a gene-specific primer. It will attach all of that into a single amplicon, and then that amplicon becomes the Illumina-ready sequencing library. Mentioned mixed cells or doublets. So that's basically a Poisson distribution where we have diluted out enough bead to cell ratio so that you have a very low doublet rate. Typically it's sub 10%, but that depends on the panel itself and the number of amplicons. And if you have additional questions, I'm happy to go into further detail about that.
Great. Is it possible to apply the BioLegend panel or other DNA plus protein panels to other fields other than oncology?
That's another great question. We do have additional application areas. We have some customers which are interested in understanding latent HIV. So seeing HIV, which is reintroduced back into the genome, and then understanding on a cell-by-cell basis which cells have HIV, which don't. We have other customers that are in the cell and gene therapy space. They may be making specific cell products, and they want to understand if I made, let's say, a CRISPR edit, what percentage of those cells have the edit? Is it heterozygous, homozygous? Does that edit also have off-target edits that are going on at the same time? And then understanding, does the cell look quite normal from a cell surface perspective? Let's say if I'm editing a particular population, does the cell surface profile of that cell seem normal? Other application areas that have been a little more recent, we have customers that are branching into neuroscience. They want to understand, do they have particular sets of mutations which may lead to Alzheimer's or things like that. So yeah, we have a number of application areas people are utilizing the platform for beyond just heme oncology or solid tumors.
Great. So along those lines, what is available for solid cancers?
That's great. We have a tumor hotspot panel, which is basically a panel with most common solid tumor markers. We have a bunch of other pre-designed panels which are available in Tapestry Designer. You can go in and look, and we've kind of gone into the Cancer Genome Atlas and tried to identify the most common mutations across different indications and doing that. We haven't quite done the cell surface profiling of solid tumors in addition to DNA mutations, but we can isolate nuclei from solid tumors and then put that into the Tapestry system. So that's something we'll be looking at in the future, is to expand our offering with BioLegend into solid tumors.
Very exciting. So also when it comes to support and reagent development, is anything available for the mouse system?
Oh, that's a great question. Yes, we can make custom DNA panels. I believe BioLegend has a breadth of antibodies that are on the mouse side, and so that's something we can definitely do on a custom basis. I don't know if you want to add anything about mouse.
Yeah, currently we just have the human heme oncology, but yeah, mouse is completely doable. If you need it now, we can make a custom solution, and then hopefully in the future we'll be developing mouse panels as well.
Great. And I think we have another question along those lines, which is, are there any variations based on origin of cells, animal versus humanized xenografts?
Yeah, we do have customers that are doing PDX models. So that is possible. You can identify, I believe most of the antibodies in the heme oncology panel are specific to human, and I don't think they cross-react, but I'm not 100% sure, but maybe I could add to that. And we can certainly add DNA panels.
Yeah, I think most of the panel is formulated for human only. They're very specific antibodies. I would have to double check if any have overlap, but again, these can be tested in other applications just to be sure you're going to use it, like for example, flow cytometry, just to make sure there's no kind of non-specific binding, specific binding, whatever you're looking for.
Great. Another technical question, how many cells can be loaded onto the Tapestry platform?
That's a good question. So we start the workflow, if you're doing DNA and protein, you're going to start the workflow with about 1 million cells. And then you'll go through the staining process and wash steps, which is we've optimized with our cell staining reagents and then the BioLegend panel. From that, you'll end up with about 200,000 cells. And so 100,000 cells is what goes actually into the microfluidics chip, is what's loaded. And then from that 100,000 cells, you'll get somewhere between 4,000 to 10,000 cells called from the bioinformatics. And the way we've arranged that is really so if whatever you want to do, you'll have enough stained cells to actually split into two runs if you wanted to. But from 1 million, you get down to 4,000 to 10,000 cells.
Great. Anand, I think this is a question for you. Can you spike a custom antibody into the Total-seq-D heme oncology cocktail?
Oh, absolutely. Yeah, this just because it's pre-titrated doesn't mean you can't add more or make your own panel. So yeah, that's absolutely doable.
Great. So then along those same lines, do you provide services or support for designing custom panels or custom oligos?
Yeah, yes, we do. Mm-hmm.
Yeah, and on the DNA side, as I mentioned, we have software that's on our website. You can go and create a panel on your own. Right up to, we also have what we call a white glove service, and we will have a team of bioinformatics help build a panel for your specific application.
All right. What is the average price per cell for AML panel in single cell DNA mode only?
Single cell DNA only price per cell? I'd have to look that up. I believe the pricing for AML only is about somewhere between 1,500 to 2,000 per patient sample.
Great.
So just divide that number by however many cells you get out.
How many lanes are on the Tapestry platform?
It's a great question. It's currently in the mode, it just runs a single patient sample at a time. We get the question all the time of how can I divide that cell sample? So we haven't enabled hashing at this point, but that's something we're looking forward to in the future, is being able to take that Tapestry run and then slice it across different cells. That's something we'll be working on in conjunction with increasing the cell capture rate in the coming year.
Great. What type of cells does the Tapestry workflow support?
That's great. Right now, the heme oncology panel is really focused around blood cancers, but we have worked with a number of different cells, like you said, mouse cells across different tissues. For solid tumors, we have a protocol you can isolate nuclei from, and so that works across a broad range of cell types that's possible. In general, we've done a number of different cancer types. So if you have a specific cell type question, please just reach out to us and we'll let you know if it's compatible or not.
Someone does have a very specific cell type question, which is have people run neurons?
We do have a customer. I mentioned neuroscience. They're actually running nuclei from postmortem brain. That's ongoing right now.
Wonderful. All right. Well, I think we covered a lot of ground and had a lot of great questions from the audience. Again, really fantastic product, and looking forward to seeing all the research and the papers that come out of the new capability of DNA plus protein. So thank you to you both.
Thanks, Kelly.
Yeah, thank you.
And thanks everybody for attending.